Hello, everyone, and welcome to the MMID Coding Workshop series. So our instructor for today is Petkau, and he's a bioinformatician with the Public Health Agency of Canada, and also a graduate student in the Department of Computer Science at the University of Manitoba, under Doctors Olivier Tremblay-Savard, as well as Gary van Domselaar. So today's workshop is going to provide you with a basic introduction to working with microbial whole genome sequencing data using a mixture of web-based and command line tools. All right, Aaron, the floor is yours.
Thank you, Jill, and welcome everyone to today's workshop. As Jill mentioned, I'm Aaron Petkau, and I'm going to be covering basically how to download and assemble microbial sequence data. So starting from how to search for data, and then into how to download it, and then into how to actually assemble that data into a complete or nearly completed genome. And I'll also introduce what some of these different words I'm mentioning actually mean, and as well as going over some of the software that can be used to do this. And in particular, because of some of the prior workshops, and because this is more of a coding workshop, I'm going to be introducing primarily command line tools, though there are various more graphical software or web-based software that can do this sort of analysis tasks as well. Yes, just as a reminder to participants that this workshop is being recorded and will be posted on YouTube afterwards, and to please hold your questions until the very end. And the question and answer period will not be recorded. So the learning objectives for this workshop is, well, how to download and assemble microbial genome sequencing data, divided up into a number of steps here. And then at the very end, I'm hoping to show off a data analysis tutorial I've written up, which is available on GitHub, that goes through all the different commands you need and will cover in this workshop to assemble an actual microbial genome. And so going through this code on GitHub should bring you from finding and looking at data on public archives to downloading and assembling it and looking at the quality of that genome. So starting off with what is microbial whole genome sequencing. So kind of at a very basic level, the idea is that within different organisms, we have DNA, which is essentially defining the unique characteristics of that organism, the different proteins and enzymes and other components of the cell that allow it to operate and give it its own sort of unique characteristics. And we want to essentially kind of read and figure out what this DNA contains, which can then be used later on to help us get a more clear picture of, say, what are the different genes and other components of the cell. And so for microbial, I'm specifically for this workshop focusing mainly on bacteria, though a lot of the topics I will cover can apply equally well to all sorts of different organisms, not even microbial organisms. However, some of the software I'm introducing is specifically targeted at bacterial genomes. And so the DNA itself is made up of many repeating, the actual molecule is a long molecule made up of many repeating subunits, which come in one of 4 different varieties: adenine, thymine, cytosine, and guanine. And the order of these subunits, as you traverse along the DNA strand, here, one of the ribbons you see in this image, or representation of a DNA molecule, the actual order of these different subunits is what defines the genetic code that is used to encode for different genes and other components of the cell. So genome sequencing then is taking this DNA molecule and translating it into a strings of A, T, C's, and G's, which represents the actual code that's found on that DNA molecule. So genome sequencing then, kind of in a bit more detail, is we have our DNA and we run it through a machine, the DNA or genome sequencer, and as output, we get different reads or different fragments of this genome represented as strings, normally in a text file on a computer, made up of one of 4 different characters, A, T, C, and G. But in a bit more detail, often when doing genome sequencing, you have to fragment or split apart this DNA molecule into small pieces before you can put them through the genome sequencer. So you can't necessarily run the entire DNA molecule through a genome sequencing instrument, though that really depends on the actual instrument itself that you're using. Oftentimes you have to fragment the DNA molecule into smaller pieces. And this is specifically true for the Illumina genome sequencing instruments, which is what we'll be covering in this workshop. However, there are other genome sequencers, and I've posted a video on how those work at the very end, where you can actually read larger fragments. But for Illumina sequencing, it's often smaller, often on the order of a few hundred units long is each fragment, though the size can vary. So at the very end, as output from the sequencer, you get something called reads, which are derived from these DNA fragments. So let's go into what these different reads actually are and the different sort of methods of sequencing that are used specifically within the Illumina sequencing instruments. So a read then is basically a piece of that DNA fragment that you've broken your DNA molecule up into, or a piece of many copies of that same fragment. And this read can actually be shorter than that DNA fragment itself. And so there are different methods of sequencing. 2 ones we're going to be focusing on in particular. Single-end sequencing means that you get 1 read per DNA fragment that starts from one end of that DNA fragment and sort of traverses along that fragment in only one direction. Hence the name single-end sequencing. Paired-end sequencing then is when you actually have 2 reads per DNA fragment, and each read is starting or originating from opposite ends of that DNA fragment. And kind of traverse or move towards each other as you traverse along the read. And so the advantage with paired end over single end is that you can actually read larger fragments. For example, because you're starting from each end and moving towards the middle. And also you can have essentially multiple copies of certain sections of that fragment, which can help with error correction later on. So the relative size, the DNA fragment with respect to the reads can be long. So the reads can actually be shorter than the DNA fragment, and there can be this gap in the middle. Or the DNA fragment can be short, and the reads can actually overlap with each other. So that is it for an overview of genome sequencing. Now let's go into genome assembly. So as I mentioned, when doing sequencing, you are essentially, at least for the Illumina sequencing, you are breaking up your DNA molecule into relatively short fragments, often on the order of 1 to 300 characters or nucleotides long. And as output, you're getting these reads, which are potentially even shorter than those DNA fragments. And so once you have all these reads, then the question is, what do you do with them? And this is where genome assembly comes in. The idea being that you want to essentially reconstruct ideally the entire chromosome or chromosomes or other small pieces of DNA like plasmids from these individual reads. And so one metaphor you could think of, or one comparison you can think of, is a puzzle. The reads are like pieces, and you want to fit these pieces together to reconstruct the entire picture that the puzzle is showing. So when going into looking at this with respect to genome sequencing, again, you have your reads. These reads actually could overlap with each other and likely do as you kind of traverse along the genome. And so these overlaps are maybe sort of like the ways different pieces fit together. And genome assembly then is the process of fitting all these reads together to reconstruct the original genome. And so once you fit all these pieces together, the reconstructed genome may not be entirely complete, but the larger fragments or larger pieces of this genome you get from a genome assembly process are called contigs, for contiguous fragments of the genome. And there's a few reasons why you may not get the entire chromosome as one long string, but one of them being that the different genomes can have repeat regions, or essentially duplicate copies of the same DNA in multiple locations in the genome. And this makes it difficult to essentially unambiguously reconstruct the entire chromosome all as one long string. And so often you'll get smaller pieces of the genome where there's essentially repeat regions in between these pieces, and you don't really know how these fit together beyond that. So there's two different types of genome assembly you can do. One is called de novo, and another is called reference guided assembly. And so the kind of main difference between them is whether or not you're using a reference genome as guidance to help you reconstruct the original genome. And if you kind of go back to that puzzle picture I showed, reference guided assembly could sort of be thought of like having a picture of the original image that your puzzle is derived from as a guide or as help to rebuild the actual genome. Whereas de novo assembly is like not having the picture of the puzzle. And so that can be useful, but one difference between puzzles and genomes is that you often don't have an exact picture of the genome available to help you reconstruct the original genome. That's sort of what you're trying to build through genome sequencing and assembly. So you often have to use, if you're using reference guided assembly, a closely related genome that you hope is not different enough from the genome you sequenced. And so that's one disadvantage of using reference guided assembly, is that the reference genome you use, if it's too distant from the genome you sequenced, can potentially bias your results. However, de novo assembly, because you're not actually using a reference genome, you're just using the DNA fragments themselves and looking at how they overlap and how they fit together. You're not necessarily biased by any other outside source of information. However, the disadvantage is often you can only reconstruct smaller contig fragments from a de novo assembly process. So for genome assembly, for this talk, I'm going to be focusing on de novo assembly, where we're not using a reference genome as a guide. So the overall purpose of genome assembly is, well, the kind of primary purpose is to reconstruct the original genome from your reads. However, what you actually want to do with assembly or with genome sequencing in general is, once you have the genome, you can then look for interesting genes or other components of interest in that genome. So you can look for antimicrobial resistance genes that can help you determine whether or not this particular bacteria is resistant to different antimicrobial drugs. You can look for genes related to different typing methods of bacteria, such as serotyping. You can also compare lots of genomes together to reconstruct phylogenetic trees or family trees of how different genomes relate to each other. And one example there that's kind of in the news is the different variants and different types of the SARS-CoV-2 virus. And while that's a virus and not a bacteria, phylogenetics and genome sequencing are very similar between the two, in the sense that you can get strings that represent the actual genome and then compare these genomes together to kind of look at how, look at the evolutionary process of either this virus or the bacteria. So now let's go into where to find sequence data. So there's a number of different sources where you can look for sequence data, but there is actually one large international collaboration that archives nucleotide sequences called the International Nucleotide Sequence Database Collaboration, or INSDC. And this provides free and unrestricted access to nucleotide sequencing and other data that is stored across different institutions spread out across the world. So this would be stored at the DDBJ, the DNA Database of Japan, ENA, the European Nucleotide Archive, as well as NCBI, I believe the National Center for Biotechnology, and I forget the I, but anyway, these different data centers are distributed across the world and archive a nucleotide data and provide it as sort of open access for anyone to use. So these institutions mirror each other, so you can actually access the same data from that's stored at NCBI from ENA, or that's stored at ENA from the DDBJ. Though I don't know the exact timing of how often these their mirrors are kept in sync. However, you can always go or kind of switch between different institutions or different websites to look for the same data. And there's actually lots of data stored and mirrored across these different institutions. So there's on the scale of petabytes of data that are stored right now, just of nucleotide sequence data. And a petabyte is around 1,000 terabytes or around 1 million gigabytes. And this chart shows about, 16 petabytes total as of, I think that was 2020. And it just keeps on increasing over the years. So for this talk, we're going to be focusing primarily on tools and the NCBI website for searching for nucleotide sequencing data. However, again, because the data is mirrored across these different institutions, you can always search for the same data at the ENA, for example, which may be useful if you find their tools easier to work with. So the primary place where you'd find microbial genome sequence data is the NCBI Sequence Read Archive, located at the website linked at the bottom. And if you go there, you'll find this page that it gives you a lot of different documentation and has a search bar at the top. And so the easiest way to search for data is to just use that search bar to search for, say, a particular organism or other interesting or other keywords you may want to look for. And you'll be able to get a whole bunch of results from this website. However, if you had a particular study or paper that references or that has its data archived at one of these nucleotide archives, it will likely report a unique identifier for this data, and then you can search for this identifier at NCBI and find the data that way. Or at NCBI, there's specific targeted repositories for certain types of data. So for example, the pathogen detection repository is targeted at DNA sequencing from bacterial pathogens. And not only does it store the original sequencing data that you can download, it also stores some of the processed data, such as storing what antimicrobial resistance genes were detected, or storing phylogenetic information about these different pathogens, which might be more useful to work with as well, depending on what sort of work you want to do. However, you can also go and re-download all the original sequencing data and run your own software to kind of regenerate or integrate this data into your own data set and results. So another targeted repository is for SARS-CoV-2 sequence data. And this not only does it contain the assembled genomes, which is kind of the sort of data a lot of people work with when building phylogenetic trees, for example, but it also will contain a lot of the sequence data related to the SARS-CoV-2 virus. And again, some of the software I show is specifically targeted at bacteria, not necessarily viruses, but a lot of tools I'm showing could be reused for viral DNA or other organisms as well. So the overall way that data is structured at NCBI within the sequence archive is the top level, you have something called a bioproject, which is meant to contain a collection of related biological data for some particular project or purpose. So for example, you can have one bioproject per study that someone publishes or per some other kind of ongoing collaboration. But the idea is that it groups large collections of genomes together or other biological information derived from, or other information derived from biological samples. And then underneath a bioproject, you can have multiple biosamples. A biosample is a record of some biological isolate derived from some source. So for bacterial related data and bacterial sequencing, it could be some particular biological or data derived from some particular colony of a bacteria that's grown on some plate. And then underneath the biosample, you have an experiment, which is, well, for sequence reads data, is related to some unique sequencing results derived from that biological data. And then underneath, so for example, a particular sequencing, if the data was sequenced with a particular sequencing instrument, that could belong under a single experiment. And then under an experiment, you have a run, which is just a record of the actual sequencing data as output by a single sort of run or single run of a particular sequencing machine. And so for sequence data, we're focused specifically on the experiment and run records in the NCBI sequence read archive. So let's look at SRA Experiment Record that you'll find if you search for sequence data on NCBI. You'll get to a page that looks like the following at the right. And so this contains, not only does it contain a description of the study that this data was kind of related to, it also contains a description of the library information, that is the way that the DNA was prepared prior to sequencing. So in this particular case, what's important for us to note is that it was sequenced using an Illumina MySeq instrument, and that it's paired ends, which means that the DNA fragments that were generated were sequenced from both ends. And so each fragment has two different reads that are associated with it. And then in this experiment record, you'll have references to the other sort of data within that sequence read archive data model. So you'll have links to the bioproject the experiment belongs to, the biosample, as well at the very bottom, you'll have a reference to the actual sequencing run, which is what stores the actual sequence data that we want to download. So if you click on that sequencing run instrument, you'll get a screen that looks like the following. And again, there's a lot of information in here that we won't necessarily look over. But well, at the top here, and I don't know if you can see my cursor, but at the top, there's these different tabs: metadata analysis, reads, and data access. So if you click on the reads tab, you'll actually see the individual reads that are found within the sequencing run. And kind of in the middle screen, you'll see these two strings of A, T, Cs, and Gs. These are actually the two different reads that were derived from a single DNA fragment. So the top one would likely be from the top one will be from one end of the DNA fragment. The bottom will be from the other end. So how to download this data? Well, if you kind of go back to that NCBI sequencing run record, you'll see another tab called data access. And if you click on it, it actually gives you a reference to this thing called the SRA Toolkit, which they recommend for downloading data. And then below that, they give you a bunch of information about how to work with this data in cloud-based environments, but that will not cover that for now. We'll mainly be focusing on using this software called the SRA Toolkit. So the SRA Toolkit itself is essentially a suite of command line tools for working with data from the sequencing read archive. And in particular, we're focusing on these two command line tools, fastqdump and prefetch, which allows you to download data and convert it to formats, mainly the fastq format that are used by many downstream tools. So the way to work with these tools, the SRA Toolkit, is to first install it. And if you recall from the previous workshop last week, we covered how to use Conda to install tools. And so the SRA Toolkit is actually available in Conda. And so you can install it similar to how it was shown last week, using Conda create to create an environment with the tool, and then Conda activate to activate that environment. And then once you're in that environment, you can then run some of the commands available in this tool. So in particular, prefetch is one command, and you can just run prefetch -- version to just verify that the actual tool installed. It should print the version of the tool that you have installed using Conda. Once you've installed the SRA Toolkit, you can now start downloading data. And so at the top there, I give you a link to GitHub, where the NCBI GitHub wiki, where it describes how to use these tools in more detail. But the prefetch commands can be used essentially to download SRA data using identifiers. And so if you recall from that experiment record at the right, there was this run section, which had a run identifier, something that starts with an SRR number. And if you just pass that or give that to the prefetch command when you're running it on the command line, it will go and download that particular sequencing run data to your computer. Once it's completed downloading, you can use another command called SRA path and use that same record, and it will actually show you where that data is located. So if you actually have it downloaded, it will print out a path or a location on your computer where that data is downloaded, which can be useful if you wanted to know where the data is, or if later on you needed to delete or clear out this data because it was taking up too much space. And if a download actually fails in the middle, you can just rerun the same prefetch command with the same identifier, and it should pick up where it left off. However, you can also use, you're not restricted to using just the SRR identifiers, the run identifiers. You can also prefetch an entire bio project, and it will go and search through this bio project, which, if you recall, contains many samples or biosamples, which contain many experiments, which contain many runs. It will go and download all that data to your computer automatically. However, you want to be careful when running this command on large bio projects, because sequencing data itself can be quite large, and if you're downloading a lot of it, that can take up quite a bit of space. So for example, this particular bio project, if you look on the NCBI website, shows you at the bottom there how much data is stored in this bio project. And so if you ran this prefetch command, it would download around 25 gigabytes of data, which might take a while depending on your internet connection. And you also have to make sure you have enough space on your hard drive to store all that data. So once you run prefetch, this downloads data in essentially NCBI specific formats. If you look back at this SRA path, you'll see the file it shows ends in .sra. This is a unique or a specific file for NCBI's storage purposes. And to actually work with it with later software, you want to convert it to more standard formats that many tools use, one of them being the FASTQ format. And so you can use a tool called FASTOrqdump to convert a sequencing run to a FASTQ format file. So you can, and it's called FASTOrq because there was an original program called FASTQdump, and then FASTOrq is faster than that original program. But similar to prefetch, you can just run FASTOrqdump and pass in the sequencing run identifier, and it will automatically convert it to FASTQ formats. And for paired end data, you'll actually get 2 files, 1 per each read pair. And by default, it will leave these files uncompressed. And you can see from this little screenshot, each file is quite large. It's about 405 megabytes. So the total space for this one sequencing run is close to 1 gigabyte of uncompressed data. If you want to save on space, you can compress it using a command called gzip, which is a particular compression format, a compression method. And you can just pass the different files, or if you're working on the command line, the star or asterisk character can be used as sort of a wildcard to represent any files that end in .fastq. And this will go and automatically compress those files and give you a .fastq.gz file. And if you see from the file sizes here, the size is about 1/4 to 1/3 the original. So now, instead of 1 gigabyte total data, you're now down to maybe 1/4 of a gigabyte of data that's stored. And so for one file, that may not be too bad to store the uncompressed data. But if you were downloading an entire bio project, that may be quite a lot of space savings, like 100 gigabytes down to 25 gigabytes, which is quite a lot of space saved. So I mentioned this format called FASTQ. So let's go a bit more in depth into what this format looks like, just so you get an idea of what the data we're going to be working with looks like. So the FASTQ format essentially stores sequencing reads it's essentially a text file that stores sequencing reads, where one read record is stored across 4 different lines in that text file. So this right now, I'm just showing an example of single end sequencing data. So you just have one FASTQ file because you have one read per DNA fragment. And so the very first part or very first line of each record is just an unique identifier for each read. The second line is the actual nucleotides that come from that read. And so this is just a string of A, T, C's, and G's. The third line is a separator that separates the nucleotides from the next line. And it may or may not just repeat the same read identifier again, or you could just see a plus sign there. The plus sign is always has to be there, but any text after that is optional. And then the very last line is the actual encoded quality of each nucleotide. So each read, in addition to having the nucleotides, the A, T, C's, and G's, also includes information that help you determine or help a computer determine how much confidence you have that this particular nucleotide is correct. And so this is encoded as these characters here. And so this maybe looks a bit sort of like a random jumble of letters and other symbols. However, it's encoded using this kind of table or this representation at the top, where each character is associated with a particular number. So for example, the very end there, the exclamation point, is associated with the number 0. So that's the minimum or lowest quality. That means essentially no confidence that that particular nucleotide is correct. And at the very right, you have I, which is the maximum quality score of 40. And so that is the highest confidence that that particular nucleotide associated with that quality score is correct. And in between, you have different characters or letters being mapped to different numbers. So A, for example, which you can see in the first two letters in this quality score line, represents the number 32. So the first two nucleotides in this read both have a quality score of 32. So that's useful. That allows you to translate that kind of jumble of letters and other symbols to numbers. But what do these numbers actually represent? While these numbers are then mapped to essential probabilities of that particular nucleotide being in error. So a score of 10, for example, means that the probability of a nucleotide at this position is incorrect is 10%. A score of 20 means 1%. A score of 30 is 0.1%, and so on. And you can read more information about how this mapping occurs and actual formula behind it at this Wikipedia site, but these are called Fred quality scores. And so these scores can help software make decisions about whether or not it should trust the nucleotides within a particular read or not. So that all was just one single FASTQ file for single-end sequencing. For paired-end sequencing, you have 2 reads per DNA fragment. And so these are often stored as 2 separate FASTQ files, where you have the first FASTQ file just has records for the first read in a DNA fragment, and the second FASTQ file has records for the second read. So now let's go into quality of reads. So to actually evaluate the quality of reads, as opposed to just looking at those quality scores and trying to translate them yourself, there's software that can actually analyze this and give you a lot of statistical information, as well as remove low quality reads so that they don't impact downstream analysis. One such software is called FastP. And so to install it, you just do conda create an environment with FastP and install the software. And to run it, you just pass the different FASTQ files. You also tell it where to save FASTQ, the good quality reads as FASTQ files. You kind of give them names, and then you also tell it what the report name should be for all the different statistical information. And so this will, as output, give you filtered reads, which contain only good quality reads, as well as your quality report. So reports contains a lot of different information, showing you kind of what these reads and what the quality looks like at a larger picture. So some important sections here are this filtering results, where it tells you the reads, the percentage and number of reads that pass filters and the reads with low quality. So in this run that I did here, 96% of the reads are good quality related to the different thresholds defined from the FastP program, and 4% of those reads were removed because they were very poor quality. You also get a lot of different charts. So this chart shows you the distribution of the different DNA insert or fragment sizes. So the insert here means the DNA fragments where the reads were derived from. And so this distribution shows you the size or the distribution of the different DNA fragments. You can see here the peak looks like around 110 for the DNA fragment size, but there's a long tail end going all the way to 500 and maybe beyond nucleotides for a particular DNA fragment. You also get another chart showing you the different quality across the reads as a whole. So this essentially just looks at the mean score over all reads as a whole. So at the x-axis, you have the position in the read, and the y-axis, you have the quality score. And this kind of shows you just over all the reads across all your data, what is essentially the mean score. And it also breaks it apart by the different nucleotides. So now let's go into how to assemble data. So genome assembly, if you recall, is sort of similar to putting together a puzzle. We have the different DNA reads derived from DNA fragments, and we want to kind of fit them together to reconstruct what the overall genome looks like. where as output, we often get these long contiguous fragments called contigs. So genome assembly, so one method to use for genome assembly is to take individual read and break them up into smaller fragments, which are called k-mers. So in this case, a k-mer where the k is just the size of the k-mer. So in this case, k is 3, which means that we're looking at basically triples or 3 triples of nucleotides only. So the read in this case has 4 characters. So we're going to break it apart into triples of characters. So ACA is 1 k-mer derived from this read, and then we shift over by 1, and we get another k-mer, CAT. And so this read of 4 characters can be split into 2 k-mers of size 3. And then we repeat the process for all the other reads. So for this other read, we split it apart again into k-mers of size 3, and we get 2 k-mers here. Now, the next step in the genome assembly process is to construct a graph. So a graph is a mathematical structure consisting of 2 main parts. You have nodes and edges. And so graphs come up in a lot of different fields. So for example, if you think of, say, cities as nodes and roads between cities as edges, you could construct graphs of the different like highway networks across the country. Or you could think of use a graph to represent computers across the internet, where nodes are computers or routers, and edges are like links between computers, whether wireless or wired. Or you can think of social networks like Facebook or Instagram, where nodes are individuals and edges are people on your friends list, for example. And so graphs themselves come up in a lot of different areas. And for genome assembly, it's no different. Graphs are quite useful in this case. And in particular, we're going to be building something called a De Bruijn graph out of these k-mers we derived from reads. And so the idea here is each k-mer is a node. So ACA is 1 node represented by a circle. CAT is another node, and an edge represents k-mers that are essentially right next to each other or are sequential k-mers. So they're kind of different by only 1 character. And so we kind of build nodes in this graph and edges based on 1 read, and then repeat the same process for any other reads. And as we continue doing this, we get an entire completed graph where you have these long, essentially, networks of k-mers linked together. And this graph may have kind of distinct parts that are disconnected from each other, or they could all be connected to each other. But once you have this completed graph, you can then look for paths through the graph that are unambiguous, that you can kind of follow without coming to forks in the road, essentially. And these unambiguous paths can be used to reconstruct construct contigs or reconstruct larger fragments of the genome. So one software that uses this De Bruijn graph method to assemble a genome is SKESA, which is targeted at microbial genomes and Illumina sequencing data. So you can install it using Conda. You run it by just passing the FASTO files and tell it where it should save the contigs. And as output, it gives you contigs, those contiguous fragments of the genome that are larger than the reads, as well as a bunch of additional information on the assembly. So looking over the assembly information that you get as output from SKESA, you can see here, it shows you a k-mer size of 21 in this case. This will show you the graph size it built. In this case, 4.4 million k-mers. And it also shows you the number of contigs produced. So around 7,500 in this case. And to note, if you actually look at the actual output of SKESA, it's essentially building graphs using multiple k-mer sizes. So this is just 1 fragment with a k-mer size of 21, but it can go up to k-mer sizes of hundreds, for example, and use this information to essentially join contigs together into larger and larger fragments of the original genome. And this is output that you get from SKESA. So it's just long strings of A, T, C, Cs, and Gs. And then there's a separator here, which shows you the unique name of each contig. So, oh, great, we have an assembly, but how do we evaluate the quality of this assembly? Well, or how do we compare two different assemblies together, maybe produced from different software, maybe with different parameters? And how do we decide which assembly is good? So let's think of a scenario here. We have two assembled genomes where these colored bars represent the contigs, the size or width of the bars represent the lengths of those contigs. So genome A, we have genome A and B here, but just looking at the contigs kind of in this sort of unorganized pile, it can be difficult to interpret. So how do we actually go and come up with ways to compare these different genomes and decide which one is better, which assembly is better? Well, one thing we can do is we can order and count the contigs. So we can order them by size and just count how many contigs we have. And this tells us that assembly B has more contigs than assembly A, which can maybe indicate that genome A is better because it's not divided up into small fragments. But this still isn't really, it's still difficult to interpret this. You can also have more contigs if they're different organisms, for example. Though in this case, I'm assuming these are two similar organisms or two similar genomes. The next step we can do is we can essentially string the contigs one after another to find the cumulative length. So in this case, let's say both genomes are 1 million base pairs or 1 million characters long. So they're both the same length, but assembly A has less fragments, and so it looks like it's probably better. But let's try and find a way to quantify this. So there's two common values you may see and named when you're looking at assembly qualities, N50 and L50. So N50 is the contig length, such that about 50% of the contigs of the assembly belong to contigs of this length or greater, and L50 is just the contig number corresponding to N50. And so that text description is a bit hard to interpret. So if we go back to our assemblies, let's cut them in half, or let's look at the kind of assembly length as a whole and cut them in half. So we're looking at 50% because we're looking at N50. And now let's start counting contigs from the largest to the smallest. So number one is still not over that 50% line. So we'll go to number two. Now we're past that 50% line. And so the length of that contig is your N50 value. And the number of that contig, the second contig, is your L50 value. With B, we start with contig 1, 2, and we have to go up to 3 before we pass that 50% mark. So the N50 is 125 and L50 is 3. And so this gives us a way to quantify which assembly is more complete, mainly assembly A, because its N50 value is bigger and its L50 value is smaller. Likewise, you can define N25 and L25 by, instead of looking at 50%, you just look at the 25% line. Or you can look at N90 and L90 by looking at the 90% line, and so on. So software that gives you all this information is called QUAST. And sorry if I'm skipping ahead, I'm just want to make sure I get to questions later on. So to install QUAST, you'd use conda, and then you just run it and give it the different contig files that you produce from the SKESA software. And as output, it's going to give you a whole directory or a whole folder of files, but in particular, we're interested in the report.html file. And so this gives you a nice file that you can open up in a web browser and compare different assemblies with each other. In particular, you can look at the total length of each assembly, so 3.2 versus 4.8 million base pairs. You can look at the N50, so 1.4 kilobase pairs versus around 300 kilobase pairs. So the very rightmost assembly looks a lot better because it has a higher N50, and it's also longer. And I know ahead of time this data is derived from the same genome, so they should have the same length. And you can also look at additional charts. So this chart is actually a NX chart. So the x-axis is showing you either N50, N20, and 90, going from 0 all the way to 100. And the y-axis is showing you the actual like N50 value, for example, and representing this as a plot where you can compare many genomes at once. I'm near the end, but I just want to mention that I've gone through a lot of different commands in this presentation. And so if you want to actually try out this yourself, I've also included additional materials on GitHub. So you should have seen this link if you got the slides, or if you go to the MMID website and find the GitHub link that way. But the idea here is that there is an entire, if you look at this GitHub page, you'll see a link here, microbial genome assembly.ipynb. This is something called a Jupyter notebook, which is a way of storing together text descriptions, images, as well as commands, all in one single file that allows you to execute everything using special software. And so if you just click on the link, you'll actually see a preview of it on GitHub, where you can go and just kind of read through the different text. And this will describe how to run and get started to work through this tutorial. And you also see these boxes here, which show you the actual commands to run. And so if you wanted to run this on your own computer, you can just copy these commands and paste them into a terminal or type them out. And right below these gray boxes here, you'll also see some text, and this is showing you what you actually got as output, or what I got as output when I ran these commands. So see here, there's another command for installing SRA tools, and this is all what you should expect as output from Conda. And then this is running prefetch -- help, and so on. So clicking this link is great if you want to kind of copy and paste commands on your own. But I also have this link right here, launch binder. And so what this will do is this will actually launch a working example in a cloud-based environment where you can actually run all the commands yourself and try them out. So this should launch pretty quickly. You can see even a preview of the instructions right here. And the idea here is that once it launches, it'll essentially create a free virtual machine for you someplace likely in Google Cloud or one of these other organizations, where you can then go and run these different commands on your own time. And keep in mind that this is a free service that's provided, which means that the machines it gives you are pretty limited in resources, and you're also limited in the amount of time you get on those machines. So it's useful for trying out different commands and kind of testing out how this all works. But to actually run it on real world data where you want to keep it long term, you're probably better off to run it on your own computers. But if you just want to try things out, you can just click that launch Jupyter button. And now it looks like it's starting up the machine. And now you'll have this interface right here. This software it's running is called Jupyter. At the left here, you see all the files you have in a folder, and the right here, you see the notebook. So just as a very brief introduction, you can click this play button here to run each individual cell, which is highlighted in blue is the active one. And when you get to a command here, it's going to actually run the commands for you on a computer someplace in the cloud. And so clicking this button, the play button, now it shows a star right here, which indicates this conda create command is running. It may take a while if you want to work your way through all these commands, but this lets you essentially test out the entire process of downloading a genome and assembling it using SKESA and looking at the quality reports. So this is it for, actually, sorry, I'll just switch back to my slides first. Most of these slides are just what I already described. So here's a bunch of helpful resources for how sequencing works and other tutorials and other information you can read through. And thank you again for attending my presentation. And we have just a small amount of time for any questions if people have them.